welcome. We're glad you're here. We'll get rolling in just a second. Okay, I think we'll get started. It is 4:03 by my clock. So good afternoon, everybody. We're so delighted that you've chosen to spend time with us this afternoon. This is the first in our first conversation in the Probable Meets Possible series, where we are bringing together University of Minnesota scientists in bio-based fields to talk about the probable challenges we face and the possible advances that could have a profound impact on our future. My name is Holly Menninger, and I'm the Director of Public Engagement and Science Learning at the Bell Museum. We are proud to be Minnesota State Natural History Museum and a public gateway to research at the University of Minnesota. Our mission at the Bell is to ignite curiosity and wonder, explore our connections between nature and the universe, and create a better future for our evolving world. So while presently our doors may be closed due to the COVID-19 pandemic, we are determined to live out this mission to engage you and our excellent lineup of researchers and conversations that will explore big ideas and new directions in science. Conversations that will allow us to engage with each other in some future thinking about what lies ahead for our health, for the environment, and for the planet. We're so grateful to our partners in this series, the College of Biological Sciences, led by Dean Valerie Forbes, and the CBS communications team, including Stephanie Zenos and Claire Wilson, in addition to our own awesome team at the Bell Museum. Thank you to all of you who helped make this possible. All right, so now we're gonna get to it. Our special guests today are Dr. Christine Solomon, a natural products chemist and associate professor from the university's Center for Drug Design, and Dr. Mike Sadowsky, an environmental microbiologist and head of the Biotechnology Institute. Our topic today, medicinal microbes. Could microbes form the basis for new and novel treatments, maybe even cures for a variety of diseases? Christine and Mike will take us on a whirlwind tour of the state of their science and share their insights about what the future holds for microbes as a pathway to health. We'll have brief presentations from each of our researchers, and then we'll open up the floor to conversation. But first, a few technical notes. Please, as you get questions, as our conversation continues, please send those questions and comments in directly to our panelists through the chat box. Our team will be monitoring those on the back end and feeding those to me so we can make sure that your questions are heard and responded to by our guest speakers. You may also have noticed that we've turned closed captioning on. If that is something you'd like to turn off, you have the power to do that on your own screen, and you'll do that by clicking on the live script button at the bottom of your screen and then clicking hiding subtitles. We're also recording this webinar, and we'll post it online on both the College of Biological Sciences website as well as that of the Bell Museum. All right, enough of me talking. We are going to start by turning it over to Dr. Christine Solomon from the Center for Drug Design. Christine, you're up.
All right. Thank you so much. It's super exciting to be here with all of you today and to be able to talk about my field of natural products chemistry and medicinal microbes. I am a natural products chemist, and my research at the University of Minnesota is focused on the discovery of new compounds to treat mostly infectious diseases in humans and in animals and in agricultural systems. But today I'm going to really just give you an introduction to the field of natural products and do a little bit of speculation about where the field is and where it might be going. So I'd like to begin by defining what a natural product is. So natural products are also known as secondary metabolites. These are compounds that are made by essentially all living things. They're made by bacteria, fungi, plants, animals, protozoans. There are more than 250,000 natural products that have already been described in the scientific literature. These are compounds that are not necessary for basic cellular life, but they're thought to benefit the organism as a whole. And to better explain this, I want to walk you through three specific examples. And the first is a natural product that many of you have probably already consumed today. And that is caffeine. Caffeine is a natural product that's made by a number of different plants. It's an alkaloid natural product that's bitter in flavor. It's probably made by the plant to deter herbivores from eating it. Another really well-known natural product is penicillin, which of course is an antibiotic, and it's made by fungi. The last natural product that I want to use as an example is one of my most favorite chemical compounds. This is a compound that is responsible for a smell called petrichor, which is the smell it's that smell of black dirt, of soil, like after summer rain on a hot day. And that smell of petrichor is caused by a terpene natural product called geosmin. So these are just three chemically different examples of natural products that are produced by very different microorganisms or organisms. But today I really want to focus on natural products that come from microbes. And there are a few different reasons for that. And one is that microbes are a historically validated source of really important natural products for clinical use. More than 25,000 natural products, bioactive natural products, have been isolated from microbes, and they're active in a variety of different areas. I'm showing just two clinically important microbial natural products here. One is a statin that was first isolated from a fungus, and the compound Lipitor is based on the statins. The other compound is vancomycin, which is an antibiotic, and it's known as the antibiotic of last resort. This compound was isolated from a soil bacterium. One of the other reasons that microbial natural products are so important is that microbes can be isolated from so many different environments. We isolate them from tropical rainforests, from the bottom of the ocean, from Antarctica, from caves and mines, from inside the guts of insects, from invertebrates, and from inside plant tissues. We essentially find microbes from every environment you can imagine. And from these incredibly different environments, we find incredible microbial diversity, large quantities of different kinds of bacteria and fungi. And what this microbial diversity means is that we also find diversity in chemistry. Now, another important concept is that these natural products are biosynthesized through machinery that's encoded in the genomes of these microbes. Most of these biosynthetic gene clusters are not expressed under a single condition. So for example, if I isolate a bacterium from the environment and grow it in my lab, I might isolate maybe 5 or 8 different natural products. But if we sequence the genome of that bacterial species, we would see that it actually has the biosynthetic capacity to make maybe 30 or 50 or 70 different natural products. And so part of our work that's happening now, and will continue to happen in the future, is to continue to look at microbial genomes, predict what compounds they might be making, and figure out ways to activate these otherwise silent or cryptic biosynthetic gene clusters. So another important concept is that these microbes in the environment almost never live by themselves. They exist as very complex microbial communities with large diversities of different microbes living together. And they are communicating with each other, they're signaling, they're using antagonism to protect their nutrients or their space. And we now have some of the tools to start to interrogate these complex communities. So we can think about how and why these microbes are living together, why and how they might be making different kinds of chemical compounds. And we can use sequencing technology to sequence them without even growing them in the lab. And so this is already happening. And we are just getting larger and larger amounts of data from these incredibly complex genomes and these complex biosynthetic gene clusters. And through this work, we can also use ecology to think about ways in which we might activate those silent or cryptic gene clusters to try to express new bioactive natural products. related to this work is the idea that we're building so much new data. We're sequencing huge numbers of bacteria and fungi. We're generating biosynthetic gene sequences. And we really need the help of computational scientists. And I pulled out this quote because I think it's really wonderful that the prospects of computer scientists working more closely with bioengineers is salivating because there's so much potential to be inspired by nature and then to build on that capacity that nature has shown us. And so I have two examples here that I think are really wonderful for further reading in this area. The last example that I want to share is based on a headline that was popular maybe a few years ago, and the idea that bacteria might have antidepressant qualities. And that's a really exciting idea. And this headline was based on some research that showed that this group took a soil bacterium called Mycobacterium vaccae. They killed it and they injected it into mice. And they found that those mice experienced lower levels of anxiety and had decreased inflammation. And just last year, that group published a paper showing that activity was in part caused by a small molecule. And that small molecule is a fatty acid shown here. And so this group is now trying to use this information to see if they could develop a kind of vaccine for anxiety or to treat patients who have PTSD or other anxiety disorders. And so that's another example, I think, that really demonstrates where our field is going. I think it's pretty easy to think about how and why antibiotics are made in the environment. If microbes are producing things to antagonize a potential competitor, that makes sense from an ecology standpoint for why we would look for antibiotics like that. But there are many more human diseases, animal diseases, and agricultural diseases that have a real need for new treatments. And so I think that natural products is going to grow in the areas in which we can find clinically relevant areas for new therapeutics. So I know that I covered a lot of material in a really short amount of time, but my take-home message is really that the microbial world is huge and diverse and largely unexplored. Most bacteria and fungi in the world have not been cultured. They've not been grown. They've not been sequenced. And within each of these diverse and numerous microbes, there's the capacity to make very large numbers of bioactive, incredibly complex, and possibly therapeutically useful natural products. If this topic is interesting to you and you want to learn more, I want to direct you to this 3-minute animation that was a collaboration that we did together with a company called Minute Earth. They worked with the University of Minnesota and they develop all kinds of animations to explain very complex ideas. And they have this one animation that explains more about how natural products are made in the environment and how they've been developed into clinical drugs. And so if you do a search for where do our drugs come from, it's on YouTube. And then I also know that there are a lot of students in the audience. And if you have an interest in this field, I want to direct you to the American Society of Pharmacognosy. As you can see here on this slide, there are lots of related fields that work in the area of natural products chemistry, and there's a real need for people in all these different areas. There's a link at the bottom if you want to learn more about opportunities for doing research in both academia and in industry. And with that, I will end here and happy to take questions.
Thank you, Christine. I'm imagining everyone at home is giving a round of applause, even though we can't see them. I'll encourage folks that if you have questions for either one of our researchers today to go ahead and type those into the chat window. I'll ask Christine like one brief question before we move on to Mike, but I want to make sure that we're capturing those questions as they're emerging. And so I guess my question before we transition to Mike, Christine, is so with so many natural products out there and all of these potential diseases or conditions that could be targeted, how are researchers knowing what to test things for? Like, I'm thinking about that example you just shared about depression and the mice and that natural product that from the soil that had an effect. How do researchers have an inkling that they should test that out in the first place?
That's a really good question. And to answer that, for the depression study, that was actually a side observation. They weren't even looking for that activity. So they were actually trying to enhance the activity of another vaccine. And they found that their mice had really different behaviors, and then they linked that to inflammation. So they weren't looking for it to begin with. And I think that's actually an important characteristic of a lot of just scientists in general, that we have a hypothesis, we have ideas we want to follow, but then you have to be open to the possibility that you might see something or observe something that's new and unexpected, and you can follow that. To more specifically answer your question, it's a hard one, and it has to do with finding a balance between what you can do in your own lab, who your collaborators are. So what kind of assays are available? What diseases do you have expertise or interest in? And it's a little complicated, and it's really limited by the amount of compound we can make in the lab, because we're not doing synthetic chemistry for this particular field. And so we might only have 3 milligrams of a natural product. So, you know, do we want to test that against cancer? Do we want to test that against a fungal pathogen? We look to the literature to help guide where we might expect to see a specific kind of activity, but we don't always make the right predictions.
Great, thank you. All right, next up we will have Dr. Mike Sadowsky from the Biotechnology Institute. He's an environmental microbiologist. Mike, would you like to share your screen?
I think I'm sharing now.
You are. Why don't we put it in presenter view? You got it.
Okay. Well, thanks very much. I'm really excited to be here today. And I'm happy to follow up from Christine's really nice talk, because I'm going to be approaching this question from a slightly different point of view. And I'm going to be talking again about the power of microbes, but this time I'm talking about using microbes to cure human diseases. And I also want to follow up on Christine about microbial diversity, but I want to add an extra dimension to that, and that is microbial numbers. So my background's in bacteriology, and I've been fortunate to have a career studying microorganisms for a long time, mostly the bacteria, and I was always amazed by how many there are and how many different types there are. So one way to put this in perspective is to just imagine you're looking up at the nighttime sky, and here's a picture of our galaxy, the Milky Way. And in this Milky Way, there's about 10 to the 11th, 10 to the 12th stars. So that's 100 billion to a trillion stars in the Milky Way galaxy. And in our limited capacity to understand big numbers like that, that seems like an astronomical amount. But actually on Earth, there are more microbial species than there are stars in the galaxy. And there's about 1 trillion microbial species on the planet Earth, with some 10 to the 30 cells, which is a nonillion number. And so there's even more cells on the planet Earth than there are stars in the whole universe, not just our galaxy. So it's a tremendously large number. And despite the fact that we've been studying them from a long time, it's only really a handful of microbes that cause disease. So of those billions and trillions, the rest are really important for us, as you just heard from Christine, that some produce important medicines and compounds. Others are engines that drive our planet, that make the cycling of elements and the recycling of materials. And others make food and chemicals and other things we need that we use every day. And what Christine mentioned too is that we really don't know a lot about the microbes. And it's been estimated that of all the microbes that we've looked at, we know less than 1% of all that are potentially around there. And I'd say that's probably 1,000 to 10,000 to even 1 million times less than we really know. And we really know just a handful of the microbes that are there. And we're way up on top of this tree of life, and we think we know a lot, but this tree is missing a lot of branches on it right now. That through discovery, through gene sequencing, and other technologies, we can fill out this tree. But for the time being, we're kind of ignorant of all the microbes there are and what they do. But I want to spend some time talking about an important thing to all of you, and that's your gut. And everyone has one. It's a very complicated organ, and it contains between 700 and 1,000 microbial species. And what's really interesting is some of the species in your intestinal tract can reach really large numbers. And one species that we all have reaches about 1 billion to 1 trillion per gram of fecal material. And that's just a small amount, because the whole human body contains about 1.5 kilograms of microorganisms. So we are essentially made of microorganisms, and we interact with microorganisms all the time. Some of those interactions are beneficial and some are not. And people have been studying the human intestinal tract for a really long period of time, and we know that the microbes that are living within you and also on you are important for your health. They supply energy, they provide barriers to disease-causing organisms, and they really are important for what's called instructing your immune system when you're really young, so that you can react to invading organisms in the proper way. So these diverse microorganisms live with us. They appear in our digestive tract shortly after birth, and they stay with us our whole life. And they're really important for how we feel and how we interact in our whole environment. And you know, if you have a problem in your gut, it makes you feel really bad, can affect your mood, your mental stability, a lot of other things. And we're just trying to understand this now with new molecular tools. So this really interesting photograph shows that the interaction between the microorganisms, which is in the top quadrant, with the human cells, which are in the bottom blue, is modulated by this membrane, this gut barrier that prevents the microbes from entering your bloodstream, but still allows them to communicate with the body through the transfer of chemicals between the microbes and the human cells. And this interaction is really important for us. Unfortunately, every so often this gets out of whack, and this is really called a dysbiosis, also referred to as a dysbacteriosis. And it refers really to a microbial imbalance within, on, or otherwise interacting with the body. And typically it's reported as a condition of the digestive tract. And sometimes it can be caused by a single microorganism that gets in the wrong place at the wrong time, or even an antibiotic that can cause this. So this whole idea that a microbe can disrupt your whole intestinal tract is not new, and we've been studying for a while, but the treatment for many of these diseases are the same cause of the disease in the first place, which is antibiotic provocation. And I want to talk to you just a little bit about this. And probably the best example of this, and many of you have heard about this, is this nasty organism called Clostridium difficile, and its Latin name means a lot because it's saying it's a very difficult microorganism. Actually, it's an easy one to get, but a difficult one to get rid of. And it's really caused due to the decimation of the microorganisms in your gut, and the gut should be looked at in this time as an organ that interacts with the body and is important for its functioning. So we had simply asked a long time ago, if microbes can cause a disease, can they be used to cure diseases? So while I'd like to take benefit, and my colleagues that works with me on this, Dr. Alexander Karatz, would like to take benefit from this particular question, if you look back in history, you find we're not the first ones to think about this. In fact, this whole idea of using bacterial therapy to treat intestinal diseases actually goes back to more than 1,700 years ago, when an ancient Chinese physician developed this material they called yellow soup, and I won't go into the details about it, but it was used to cure intestinal diseases. And this really stayed in the background of things until about 1958, when Eisman and his colleagues talked about using what they termed fecal enema to treat various enterocolitis diseases. or gut disbalances. So it's an old idea revisited. And as it was revisited, people started thinking of different ways that they can treat gut imbalances by using microorganisms, where microorganisms or fecal material containing microorganisms were placed into the human body, first through a nasogastric tube, then via fecal enemas, and as we practiced initially, colonoscopy, and more recently using capsule technology. So this is just, I should say, the tip of the iceberg. And we chose this particular disease caused by Clostridium difficile because it's a very good example where an antibiotic exposure led to the overgrowth of a single bacterium, Clostridium difficile, that produced nasty compounds by itself, toxins, that killed off all the other microorganisms in the gut. And that led to this massive dysbiosis, the worst disease that I really want to see anyone have, and a possible way to treat this disease. So we call this type of treatment intestinal microbiota transplantation. And we have started here at the university a microbiota therapeutics program that really galvanized around this particular set of diseases to begin with, and now has expanded to other diseases. I'd like to mention that I am not a physician, and I'm not a medical microbiologist. And my background is actually in environmental and ecological studies, microbial ecology. But this environment is just another example where microbial ecology can be studied in its entirety. So the US government, especially the US Food and Drug Administration, was very interested in our earlier studies that we began around 13 years ago, where we proposed to use these fecal microorganisms referred to fecal microbiota to cure diseases. And at that time, the federal government didn't even know what to call these things, because traditionally it's been drugs or chemical compounds that have been synthesized or natural, as Christine said, that have been used to prevent diseases. But they decided that the legal definition of these compounds were as a drug and as a biological product. And we followed through on that in our production of these materials, both for our own patients here in Minnesota, which is now reaching some 800, as well as those in national trials around the United States. So we started out using these different formulations. Back in 2010, or 10 years ago, we had produced this frozen material that we could put in these containers in tubes or in pouches. And then in 2015, some 5 years ago, we developed capsule technology, where one can just simply take a capsule in their mouth and cure this particular disease. This intestinal therapeutic program goes from good manufacturing processes and development to produce the drug compounds, to using a very sophisticated program of standard donors. And then we have microbiologists, geneticists, genomicists doing microbiome analyses to see how these microorganisms interact in the new host. And then from that, we derive a whole other program in translational research involving technicians, researchers, and physicians around the United States. I'd like to say that this research can't be done in a vacuum, and it costs some money, so we're fortunate to have a lot of nice donors. We hooked up with this really interesting organization called Achieving Cures Together, which is run by some of our colleagues now that actually provide funding for us, so we're really grateful for them. Also, the Hubbard Broadcasting Foundation has supported this research, as well as funding from the National Institutes of Health, various University of Minnesota granting agencies, as well as the Academic Health Center. I would like to point out towards the end here that these types of analyses and these types of pharmacological uses of these materials requires a very large team of individuals, and many of those are listed here, and the list is even expanding more. We started first treating Clostridium difficile disease only. That has moved into areas of trying to treat various irritable bowel diseases and irritable bowel syndromes. And I'd say the most exciting area we've moved in more recently has to do with autism spectrum disorder. Not that these particular microbes cure autism, but the spectrum disorder has with it a range of various maladies that can be cured, or at least the symptoms can be relieved using some of these technologies. And we learned very early that a lot of children with autism spectrum disorder have intestinal irritable irritabilities that cause them to behave poorly, and that treatment with some of these microorganisms results in their calming effect upon their behavior. So again, it's getting into issues that Christine probably meant. So this is just the tip of the iceberg. I'm really happy and I'm fortunate to be involved in this early research. And as you can see, in this case, we're treating diseases using microorganisms only, no drugs. So with that, I'd like to end my discussion and pass the baton back to Holly.
All right, thank you, Mike. Everybody at home or in your offices are giving a round of applause. Thank you, Mike. Maybe as people are kind of gathering their thoughts together, putting chats or questions into the chat window, thank you all to folks who have submitted questions so far. Mike, I'd like to ask you a follow-up question. So you were talking about thinking about therapeutics, particularly focused on the gut. And I'm wondering if there are other researchers out there that are thinking about dysbioses in other places in the body where microbes may play a role in a therapeutic way.
Great question. So I think the two hot areas right now are the relationship of the microorganisms in your mouth. So it's the oral microbiota. And we know that some people are really susceptible to getting diseases of the mouth or the teeth. And people are looking at whether they can transplant microorganisms from a good mouth microbes to bad mouth microbes and see if they can change them. And the other area of really interest right now is on the skin. And we do know that there's a lot of skin abnormalities or dysbioses caused by either your own personal chemistry or the environment you live in. So those are two really hot areas now. And then there's this other really interesting area which kind of links the brain to the gut. And that's called the gut-brain axis. And we all know, as I said before, when your gut's not doing well, you feel bad. Or when you're feeling bad, your gut doesn't do well. So there's a communication, and people are really interested in studying the gut-brain axis.
Awesome, thank you. Christine, if you'd like to unmute too. So my hope is that we have really a conversation between the three of us and the 146 people that are watching online. And maybe one thing to kind of tie your two presentations together that just occurred to me, and as we're talking about these different communities of microbes and places where microbes may have a role in therapeutics. Christine, you presented photos of lots of different places where you're prospecting microbes to make natural products. Has one of those places been the human body at all? Or are folks in your field looking at the microorganisms on and around the human body as a place or potential source of natural products?
I have not done any bioprospecting of the human body myself, but I know that that is being done by a number of other labs. And the body is an amazing environment for microbial diversity, as Mike also alluded to. I know that there is some really interesting work also being done with skin biologicals, so which Mike also mentioned. And so we know that there are some protective microbes that live on the skin that can inhibit or prevent the growth of like MRSA, so Staphylococcus aureus. And so, and maybe those people who have those microbes are less susceptible to those infections. And so trying to identify those microbes and really understand how they work is definitely being done in other labs, just not by me.
Yeah, and it's interesting. Even though I said there's a large number of organisms in the human gut, so it's 700 to 1,000 species, that's dwarfed by the number in the soil. And it's thought to be 10,000 species in a gram of soil. So people have typically gone to those types of environments because there's a lot of diversities there. But I think that's a great question.
Mm-hmm. Awesome. So another question, I think that for both of you, that it seems like advances in biotechnology, so whole genome sequencing and high throughput sequencing has really allowed, I think, both of these fields to leap forward in a pretty short period of time. I'm wondering if you had your magic crystal ball, what do you think, like what's the next big leap in your respective fields, or like what's gonna just like take us to the next level?
Christine, you want to go first? I can go first. I don't care.
God, I know you, like, what are your magic 8-ball? Like, what is it telling you?
So the genomics era that we live in now has been unbelievable and has really changed science as we know it. But what it's really good at is telling you who is there, not what are they doing there. And it's really easy to tell who is there by gene sequencing. But the function of those microbes, I call the Holy Grail, because that's what we need to figure out. So as the technologies evolve and get smaller and smaller and smaller, we'll be able to start looking at some of those things much better than we have now.
It's hard for me to imagine like a huge leap in technology, because I feel like so much of what has pushed this field forward has been incremental but rapid advances all on top of each other. I also think that this incredible coming together of seemingly different fields, so computational chemists, ecologists, biologists, and microbiologists doing this incredibly data intensive work has been phenomenal. And I had that one slide showing the collaboration with engineers. So engineering things that occur in nature and imagining how we could change them and improve upon them and adapt them to our own uses. I think that's huge, and it's being done now. So that's not a huge leap, but I think that it's going to happen more and more and be more accessible. It's going to be cheaper. So sequencing costs have come down tremendously. It's faster. And so we can ask more and deeper questions and answer them using all these technologies together.
Mm-hmm.
I think one of the things, I think one of the things that Christine alluded to is this concept of collaboration, who your collaborators are, because it's been a real paradigm shift over the last, I would say 30 years even, when people work by themselves in their lab. And I know that probably some of our people think, though, there's this old guy in a lab coat working in his basement discovering something. Well, those days really aren't around anymore, because to make these big leaps or even small incremental leaps, we all need to work together as teams. And even in my own work, I work with pharmacologists, I work with genomicists, I work with people that can formulate things, I work with companies that manufacture the types of capsules that I want to use. It's really broad interactive collaboration, and that's where we're going.
Okay. Speaking of collaboration, one of our viewers today, Douglas, has asked about, maybe he's showing us cards about how do you see a data scientist assisting in your research?
Oh, that's interesting. So actually, when I started doing some of this, what's called metagenomics, which is where you take an environmental sample, extract all the DNA and sequence everything, there really wasn't anyone here that could help me. So what I did was started to learn how to do it and had people work with me that learned how to do it and became what's now called computational biologists. So there's these people that really focus on dealing with this big data and trying to parse out something that's linked within it. And working with data scientists is obviously the future where we go. And the other future is, how do we retrieve all this information? We know how to store it, but how do you retrieve it, look at it? And as the data becomes bigger and bigger and bigger, it actually becomes more difficult to do so. So we need people that know how to write databases and how to query them and how to retract the data and how to analyze the data and people how to generate the data. So it's amazing what we need to do.
Yeah, it's really critical. And another piece that I would emphasize is tool building. So I collaborate with data scientists who are really good at building tools that make the data accessible, that make it so that a biologist or a chemist can really look at the data and make sense of it. Because a lot of data that is not in a form that you can understand is useless. And so having data scientists be able to build tools that can really bring together all the people that need to understand that data is really important. And there are people that are really good at that.
And then there's one other group of people that we tend not to talk about, but we should, because as scientists, we're usually not the best communicators. So there are people around like Stephanie and others that are science communicators. They're helping us when we take all this stuff and figure out what it says. how to tell people about it so they understand it.
Yay, I'll give a shout out to all the science communicators. Well, I'm looking at all of the questions coming in, and it seems like our audience is really eager to make some ties between our current situation with the COVID-19 pandemic. And so I have a few questions from multiple people that are kind of exploring those different dimensions. One is from Anna, and she says, I've heard dysbiosis might be a contributor in those who develop worse symptoms from COVID-19. Do you know if this is being investigated?
I personally don't know of anyone investigating this right now. In fact, I'm a little ignorant of the area, to be honest with you, because my background is in bacteriology, and I don't have a background in virology. And I think you need two of those people to come together to answer that question.
What about another, Kipling asked about microbes in the lungs, and maybe that falls in that same category if there are people that are, regardless of whether it's related to COVID or not, like investigating what might be going on with the microbial communities in our lungs.
Yeah, so that's a really great question. When we used to think about the human body, when that was born, that you come out sterile, and that you're rapidly colonized by microorganisms, chiefly or initially by those of your mother and then of your surrounding environment. And as life goes on, those microorganisms change as you impact them, and that areas that we used to think were sterile are not really sterile, and that one of those areas are the lungs. That's called the lung microbiome. There's also folks that believe that there's a small circulating microbiome as well. I just caution people that when we start looking at these things, it's sometimes difficult to separate contamination from when we go into the site and remove a sample. So I think that's up for review still, that we know there's microbes in the lungs. Whether they're permanent residents in the lung is a question, and what they're doing there is another question. And the same with the circulating microbiome.
Christine, I think this one is more on your alley about whether bacteria and fungi produce antiviral compounds, and are these being investigated?
Absolutely. So there are lots of natural products that have shown activity against a variety of different infectious viruses. I know there's a question about COVID-19 as well, and I would imagine that there are people that are actively screening their collections and libraries of natural products against the coronavirus. The challenge there, of course, is finding a biologist or medical collaborators who are growing that virus and are able to test them in a format that is accessible to you with your library. And so some people have libraries of compounds that are available that they can just ship out. And I'd bet money on that actually happening right now. I don't have any collaborators at this moment who I can send my compounds to, but I think that in a very short amount of time, that's going to become available.
That's great. I guess I have a COVID related kind of question, and it's less about like addressing it directly, but more about like what's happening with the process of science. Like we hear about in the news that scientists and doctors have mounted this massive rapid response in terms of their science, whether they're looking for antiviral drugs or vaccines or ways to test for the disease or or antibodies, and motivated by this enormous sense of urgency. And as a result, I think we're seeing that like the fundamental way that science is being conducted and how it's being communicated too, because we're hearing about research that's coming out before it's gone through the peer review process. How does that kind of rapid response way of doing science, how do you think that will affect your own field looking ahead into the future? Will it have a lasting impact on the process of science writ large? Maybe that's too big of a question.
No, I can take a stab at it, and then I'll let Christine respond as well. So this type of response, I should say, is not new. And we've seen this when there's been several different outbreaks of diseases or several different things happening to the country at one period of time. And I look at it as a small perturbation in the way that we normally carry out things. And I'm not saying that way we're doing it now is not bad. People are really just, I should say, really anxious to get something that works. And so we're talking a lot about it. I think the thing that we have to just keep in mind always is what's the best thing for the patient, what's going to actually help the patient and not harm the patient. And those things have to always be in your forethought when you're doing things.
Yeah, so the other disease area that I work on is Whitenose Bat Syndrome, which is a fungal disease of bats. And we're working on looking for biological control agents to help treat this disease. And this is an example that is also very dire and has occurred only in the past decade, slightly longer than a decade. And so there's been a similar, not quite as intense, very rapid response. And it's been really remarkable to see how science has changed around that situation. So for example, once a month, there's a national phone call that most of the researchers in the field go on at the same time, and we share information essentially in real time. And that has not really happened in any of my other areas of research. You sort of wait till you have a polished, finished piece of information that you publish, and then you present it maybe at a conference. So this has been a different experience. I think it has to be done very, very carefully. So as we've seen some examples where information has been shared with news agencies, there have been misinterpretations. That vetting process and that peer review is super important. But we're also in a situation where we need communication to happen really fast. And so finding a balance there is hard.
Yep. We have, changing gears a little bit, a question from Anna. She asks, how much influence does soil bacteria affect our microbiome? How much has soil bacteria changed with the advent of industrial farming? That's a very big
that's really interesting
set of questions.
So there are actually people that study this, but they study it more from not soil bacteria just in the ground, but those that enter the atmosphere and interact with us. And there's folks that study the indoor environment and how that impacts your microbiome and how you impact the microbiome of the environment around you. So there is an interchange between the microbes in the environment and your body. Some of them end up being really good for you, and others, it's not too good. And we have allergies and things like that. But it's a really interesting area. As you know, we're an integral part of this planet, so we interact with everything around us, including all the microbes and in the soil, the water, and the air.
Christine, Leslie asks, how do you and other scientists target certain environments or regions to look for natural products for therapeutic use? Do you just look everywhere, or do you have certain locations, or do certain locations tend to yield more natural products?
That's a great question. Well, and speaking for myself, I try to think about where microbial interactions are important, and particularly where there might be nutrient limitations or where there might be competition. So the main places that I collect microbes is from underground. So we collect microbes from caves and from underground mines, where there's generally low nutrients. What nutrients are there are probably limited. You could imagine that microbes are fighting for the nutrients that might be available. But honestly, I could isolate brand new microbes from the bottom of your shoe and find new molecules. So it's also just a personal preference about what environments you really want to deeply understand, what ecology you're interested in, what kind of ecological questions you want to ask. So I think it's a complicated answer.
Kelly asks, are there opportunities for students to somehow get involved in these projects? Whether places they can go for learning, which I think you shared a few examples, Christine, or ways they might help or connect to the research.
Yes. Yeah, so having students interact with our research is really central to the university's educational goals. And we allow students to interact with us and work with us, both as undergraduates, as graduate students, and as postgraduate, as technical support for our labs. Most of the colleges that we interact with, including the College of Biological Sciences, are very interested in having students work in our laboratory. And those can be done by directed studies projects, directed research projects, and also various internships that occur during the summer. So it's of interest to CBS and to other colleges to really foster those early interactions between students and science.
Yeah, I'd say particularly for students that my research is absolutely dependent on students, and I frequently have many undergraduate students working in the lab in various capacities. Obviously, it's much more challenging and difficult during this quarantine period, although we're hoping to gradually and safely get back into the lab. Some labs working in my field are much more data heavy, and so I can imagine that there are opportunities to work from a distance, doing computational analyses, building tools. In my research program, we do most things empirically, so we need to be in the lab, growing things, purifying things, looking at structures, taking measurements. And so hopefully I can bring on more students when it's safe to do so. But I would encourage anyone who's interested in this to really reach out to faculty, there might be companies that are doing research in this area, and there are definitely opportunities to do directed research or internships.
That's great. One of the topics that have come up a couple pieces in the questions are about antibiotic resistance, and I think that's increasingly becoming this really huge public health concern, especially as we're hearing like our arsenal of drugs is limited. And so I was wondering if both of you might speak to where your respective fields are offering promise in that area.
Well, I'll speak first because Christine knows more about this because she's actually working on development of some of these drugs. But in my particular area that I'm talking about now, where we're using microbes to cure diseases, it's done in the absence of drugs. And drugs are really essential to us being around. When I think about drugs, I mean antibiotics, for example. But it's also the overuse and misuse that's created some problems. So I think, like every other human endeavor, we really need to be careful of how much we use and when we use it. And if you have a common flu or a common cold or something, you just can't run to your doctor and say, give me antibiotics right away, because that leads to some of the problems. And the microbes always outsmart us in the end. So no matter what we do, we have to be one step ahead of them.
Yeah, this is a really active area of research for a lot of people looking for new antibiotics in particular, or new antifungals, because we have drug resistance in both areas. There's a few different strategies that people are using. One is to think about ways of inhibiting microbial growth without killing it. So one idea is that the reason that microbes or pathogens become drug resistant is because you're trying to kill them. And so it's this like, chemical warfare that's happening, and the pathogen is evolving and mutating and developing resistance because you're trying to kill it. And so we can think about creative ways of inhibiting growth without killing it. So for example, if we think about how certain pathogens infect the body, we can maybe stop that infection process. We can stop the production of toxins. We can stop microbes from making biofilms, which is where they grow on the surface of, for example, the lungs. And so we might be able to allow the body's natural immune system to take over if we can just help that pathogen be less pathogenic without actually trying to kill it. Yeah, so that's just one example of how we're dealing with looking for new antibiotics and dealing with drug resistance.
That's great. Since maybe we'll end on a future question. So what is the most exciting research finding that you've heard in, let's say, the last 6 months, or that your most, or maybe a research question that you're just like, yes, that's it. That's where the future is headed, whether it's for your own research program or for your field in general.
So I guess for me, the excitement comes about this new area that's opened up in understanding microbe communications. So we kind of look at microbes in the lab as individuals, and as Christine said, they live in these complex microbial communities, and they're talking to each other all the time. and just trying to understand that communication and how that communication interacts with the human host and the environment around us. To me, that's the most exciting thing.
Yeah, and I'd say my answer is kind of similar in that when we think about developing a new drug or a treatment, there's this newer context of dealing with the microbiome of the human body and not wanting to upset the good things that are happening there. And there's so much amazing research, you know, out of Mike's lab and his collaborators that are trying to understand these complex communities. And so if we're going to make a new antibiotic, then we really have to think about what it's going to do to the rest of the body and the gut bacteria and the immune system. And I'm excited about how interdisciplinary both of our fields are. And so I think there's a lot to look forward to.
Yeah. And I think on that, I think we will wrap it up so that we can stay on time. Thank you both for sharing your research, for sharing your insights. Thank you to our audience for sharing such fantastic questions. I'm sorry that we weren't able to get to all of them. Maybe we can share some of those out with our panelists so that they know what was out there in the ether that people had questions about. So thank you all. I hope everyone's giving a round of applause and shaking their hands from wherever they are. Please be on the lookout for those of you that participated today for a brief survey that will come out by email. This is our first go of this, and so we'd really appreciate hearing your feedback and suggestions. And we hope that you consider coming back next week to join us for the next edition of our Probable Possible series. It'll be on water what ifs, and it will feature Dr. Leslie Knoll and Dr. Daniel Stanton. You can RSVP via the Eventbrite links that you see. And then we also have the link for our May 27th, Not Business as Usual, with Drs. Jessica Hellman and David Bankston. So thank you all for joining us. We appreciate you being here. We appreciate you asking great questions, and take good care.
Thank you. Bye-bye.
Thank you.
Have a good night.
Good night.